The power of muscles cannot be underestimated. You have seen many real-life instances when the sheer power of muscles can allow one to flee from danger. And in some highly trained individuals, the power of their muscles can even debilitate or kill an opponent. This video mainly covers about the molecular mechanism of skeletal muscle fiber contraction. In the second half of the chapter, we will cover single fiber and whole muscle mechanisms of contractions. At the end of this video lecture, you're expected to distinguish between three different muscle tissues of the human body, describe the ultrastructure of the muscle organ and the muscle fiber, outline the process of neuromuscular transmission, and summarize the process of excitation-contraction coupling, and finally discuss roles of ATP in muscle contraction. There are three types of muscle tissues in the human body: the skeletal, cardiac, and smooth muscle. The ultrastructure and mechanism of contraction of each muscle type has slight differences. After learning about the cellular and molecular mechanism of skeletal muscle contraction, in an assignment, I would like you to contrast the slight differences of muscle contraction mechanisms between skeletal, cardiac, and smooth muscle tissues. Metaphorically speaking, a muscle is like a Chinese box with layers upon layers of smaller components, like that of the biceps brachii of the upper arm. The muscle organ, or biceps brachii, is composed of bundles of muscle cells or muscle fibers that are very long. Each of the muscle fibers are bundled up together in muscle fascicles. And each of the muscle fibers, in turn, are composed of long, thin, protein-rich fibers, which we call as myofibrils. The dark and light bands or striations of skeletal muscles is mainly because of these repetitive dark and light bands along the myofibrils length. The dark band is also described as A band, and the light bands is described as I band, mainly because of how they change the direction of light. A stands for anisotropic and I for isotropic. In the myofibril, the A band, if to be examined histochemically and under electron microscopy, is composed of a bundle of thick proteins called myosin filaments. And the I band, being lighter, is composed mainly of bundles of thin actin filaments. The myosin filaments are anchored together in bundles by a transecting network of protein called M-line. And M stands for a German term called Mittelscheibe, or the middle disc. And the I band, composed of actin filaments, is also anchored by a network of proteins at the middle called Z-line. Z stands for Scheibe. Now, during muscle contraction, what appears to happen under the ordinary microscope is that the A bands appear darker and the I bands shorten and eventually disappears in full contraction. Now, because of these repeated segments along the length of the myofibrils that shortens when the muscle contracts, each of these segments are called sarcomeres. These are the smallest contractile segments along the muscle myofibrils. So during muscle contraction, the thin filaments are described to slide along the length of the myosin filaments towards the middle of the A band. This has been described as the sliding filament theory. That is, during muscle contraction, The thin filaments slide along the length of the myosin filaments towards the middle of the A band. So the thin filaments slide along the myosin filaments. So it's called the sliding filament mechanism or theory. As shown here by electron microscopy, you have the myosin filaments that forms the dark bands or the A bands, and then you have the thin filaments forming the I bands. To better understand the molecular mechanism of muscle contraction, let's take a closer look at each of these contractile proteins in the myofibrils. Let's start with myosin filaments first. The myosin filament is composed of protein myosin molecules that has two parts, a myosin head and a long myosin tail. The myosin tails are bundled up together to form the main bulk of the myosin filament, and the myosin heads project sideways or perpendicularly. Each of the myosin head contains two binding sites. One is a binding site for actin filaments, and the second binding site is for ATP. This second binding site also has an enzyme called ATP hydrolase that can dephosphorylate ATP, thereby releasing energy for the connections made by the myosin head to the actin filaments. Because it is this myosin head that interacts and makes connection with the actin filament, the myosin head is also known as a cross bridge. It forms a connection between the myosin filament and that of the actin filament during muscle contraction. So you have the cross bridge of the myosin filament formed by the globular myosin part of the myosin molecule. The thin filaments, which are relatively thinner than the myosin filaments, are composed of three parts. They're actually a complex or composite molecule made up of a helical chain of globular actin molecules forming a bead-like helical filament that makes up the main body of the actin filament. And then you have long proteins that intertwine around the globular actin filaments we call as tropomyosin. And then the third part of the actin filament is globular proteins called troponin that has a binding site for calcium. The globular actin molecules that form the main body of the actin filament also has binding sites. These are binding sites for the myosin heads or the myosin cross bridges. So you have binding sites at the actin filament or actin molecule for myosin cross bridges, and then you have binding sites for calcium at the troponin molecules of actin filament. Normally in an unstimulated or uncontracting muscle, the binding sites of actin for the cross bridges or myosin heads is hidden by the arrangement of tropomyosin as it binds to the actin filament in an unstimulated muscle. But when the muscle is stimulated, this tropomyosin that blocks the myosin head cross binding sites of actin will be displaced such that the actin binding site for myosin heads will become exposed, allowing for interaction between myosin head and actin. This happens the displacement of the tropomyosin happens when the troponin are bound with calcium. When there is no calcium, then the tropomyosin hides or blocks the binding site of actin molecule for myosin cross bridges. But when calcium binds to the troponin molecules, this troponin molecule pulls on the tropomyosin that exposes the binding site of actin for myosin heads. The GIF image shown here shows the repetitive interactions of the myosin head of the myosin filament to that of the actin filaments during muscle contraction. These repetitive interactions of myosin head of the myosin filament to that of the actin filament is called cross-bridge cycle. It's a mechanism that repeats itself, and so it is called the cycle. And this repetitive movement and interaction of the myosin cross bridges to the actin filament involves the attachment of the myosin head to the actin filament binding site, and then the swiveling motion of the myosin heads that pulls the actin filaments to the middle of the A band, and then thirdly, the detachment of the myosin head from the actin filament. This attachment, swiveling, and detachment of the myosin head to that of the actin filament is what we call cross-bridge cycle, and it is repeated 100 times during muscle contraction that effectively pulls the actin filament to the middle of the A band along the myosin filaments. The attachment, swiveling, and detachment of the cross bridges or myosin heads to that of the actin filament is mediated by energy that comes from ATP. As the ATP hydrolase breaks ATP, it releases energy that allows the myosin heads to swivel and detach, and then to attach again and detach, and so on. So a very important component here that is not shown is that of the phosphates that are released from ATP by ATP hydrolases that are present in the myosin heads. These these bonds of the phosphate groups from ATP yields the energy that enables the cross bridge to attach, swivel, and detach from the actin filaments. Apart from the contractile protein filaments that are highly organized in the myofibrils of muscle fibers, you also have a network of membrane systems that are very important to muscle contraction, especially during the activation of the muscle. We call as excitation. These are the transverse tubules that are tubular infoldings of the plasma membrane of the muscle cell that runs into the interior of the muscle cell, running around each of the myofibrils at regular intervals and segments of the myofibrils. These transverse tubules conduct the waves of action potential that passes along the sarcolemma or the plasma membrane of the muscle fiber. And when these waves of action potential traverses along the transverse tubules, these action potentials therefore runs into the interior of the muscle fiber. Now, alongside each of these transverse tubules are a network of sleeves, or rather sleeves of tubular networks that stores calcium. This network of tubules that form a sleeve around each of the myofibrils that are alongside or uh, that flank the transverse tubules is called the sarcoplasmic reticulum. Again, let me repeat, the sarcoplasmic reticulum forms a sleeve of tubular networks around the myofibrils that flanks at each side of the transverse tubules. And more importantly, these sarcoplasmic reticulum are storage organelles for calcium. Calcium that acts as the second messenger of muscle contraction. When waves of action potential travels along the sarcolemma or the plasma membrane of the muscle fiber, these waves of action potentials runs into the interior of the muscle cell along the transverse tubule membranes. And as these wave of action potentials travels along the transverse tubules, they pass along the sides of the sarcoplasmic reticulum in such a manner that they also cause the polarizing potentials of the sarcoplasmic reticulum, thereby causing the sarcoplasmic reticulum to release, rather first to open up ion channels, calcium ion channels, that allows the diffusion or efflux of calcium from the sarcoplasmic reticulum. The image shown here shows the neuromuscular junction. The left side shows the electron micrograph of the neuromuscular junction, and the right side shows an artist's depiction of one neuromuscular junction. The neuromuscular junction essentially is made up of the axon terminals of alpha motor neurons that are in contact like a synapse between neurons on the membrane of muscle fibers. So you have in this artist depiction the axon terminal or the terminal lobe, as it is also called, of the motor neuron that is facing a part or a patch of the membrane of the muscle fiber. The axon terminals, like in synapses of nerve fibers or nerve cells, possess vesicles that contain neurotransmitters that are called acetylcholine. You have lots of mitochondria as well that provides power for the diffusion and transport of ions, as well as for the movement of the acetylcholine vesicles during neuromuscular transmission. The axon terminals of the motor neuron possesses voltage-gated ion channels for calcium as well as for sodium and potassium, so that when the wave of action potential or nerve impulses runs along the axon, they eventually end up at the axon terminal. So you have depolarizations at the membrane of the axon terminal. This depolarization in turn opens up calcium ion channels that allows the extracellular calcium ions to diffuse into the axon terminals. The opposing region of the muscle fiber is formed by a heavily folded patch of muscle fiber membrane. We call this heavily folded patch of the membrane that faces the axon terminal of motor neurons as the motor end plate. The motor end plate are rich with ligand-gated ion channels for sodium and potassium. Because they are ligand-gated, these ion channels can only open when a chemical transmitter binds to them. And in this case, acetylcholine that comes from the axon terminals of the motor neuron. As the acetylcholine binds to these ligand-gated ion channels of the motor end plate, they open up, allowing graded potentials to occur at the motor end plate, which we call as end plate potentials. Besides the ligand-gated ion channels for sodium and potassium at the motor end plate, you also have lots of transmembrane proteins called acetylcholinesterase that are actually enzymes that breaks down acetylcholine as soon as they bind with the ligand-gated ion channels. So you have acetylcholinesterase enzymes here that are numerously found at the motor end plate, enabling the transmission of signals for contraction to stop or to cease the moment they are stimulated. This allows for very fine-tuned control of muscle contraction with the presence of acetylcholinesterase that stops the depolarizing signals from the motor neurons as soon as muscle contraction is initiated. The figure here shows the process or events of neuromuscular transmission when signals coming from motor neurons are transmitted to the muscle fiber to initiate muscle contraction. It starts with the arrival of the nerve impulse or waves of action potentials at the axon terminal. These waves of action potential causes voltage-gated calcium ion channels to open at the membrane of the axon terminal. The opening of the ion channels for calcium causes extracellular calcium to diffuse into the axon terminal. And the influx of calcium induces synaptic vesicles containing acetylcholine to bind with the membrane of the axon terminal. Binding of the acetylcholine containing vesicles at the axon terminal membrane allows for the exocytosis of acetylcholine from the axon terminal to the neuromuscular cleft or space between the axon terminal and the motor end plate of the muscle fiber. The acetylcholine then diffuses across the neuromuscular cleft and then binds with ligand-gated ion channels at the motor end plate. Binding of acetylcholine at the ligand-gated ion channels of the motor end plate causes graded depolarizing potentials called motor end plate potentials at the motor end plate. These are graded potentials, but their amplitude or their strength is more than enough to initiate and evoke action potentials at the periphery of the neuromuscular junction. There are no voltage-gated ion channels for sodium and ion at the motor end plate, but these voltage-gated ion channels are more concentrated and abundant at the periphery of the neuromuscular junction, so that whenever there are motor end plate potentials occurring at the motor end plate, this travels in a summated manner to the periphery of the neuromuscular junction, where they evoke action potentials along the membrane of the muscle fiber. These action potentials then travels all throughout the length of the muscle fiber membrane throughout the muscle fiber. Now, so that contraction of entire muscle fiber happens, the excitation and waves of action potential that travels along the plasma membrane of the muscle cell must be coupled with the cross-bridge cycle mechanisms of muscle contraction. We call this mechanism as excitation-contraction coupling. The very messenger that couples the excitation events of muscle fibers to that of the cross-bridge cycle that induces contraction of the muscle fiber is calcium ion. When waves of action potential travels from the neuromuscular junction and along the muscle membranes, these waves of action potentials travels along the transverse tubules. That in turn evokes depolarizing potentials at the sarcoplasmic reticulum. These depolarizations of the sarcoplasmic reticulum membranes causes voltage-gated ion channels for calcium to open up. Because calcium is concentrated at the sarcoplasmic reticulum, this opening of calcium ion channels allows calcium to diffuse out of the sarcoplasmic reticulum. Because there are binding site of troponin of actin filaments for calcium, this calcium binds to the troponin of actin filaments. The binding of troponin to the actin filaments causes the tropomyosin to get displaced at the actin filaments, exposing the binding site of actin for the myosin heads. And with this exposure of binding site of actin for myosin heads, the presence of ATP along the entire filaments causes the cross-bridge cycle to occur, whereby myosin heads attach to the binding site of actin and then swivel and then detach in a repetitive manner, so that you have hundreds of cross-bridge cycling events occurring, pulling the actin filaments to the middle of the A band, thereby shortening the sarcomeres of the muscle myofibrils. And with the shortening of the muscle myofibrils, this causes the shortening of entire muscle fibers. And with the shortening of entire muscle fibers, you cause the shortening of whole muscle organs, and thereby you have contraction. So as we have seen here, in muscle contraction, ATP has a critical role during muscle contraction. And we can cite here 3 roles for ATP. One is that ATP provides energy for the myosin cross-bridge cycling or movement. ATP also provides allosteric binding of myosin cross-bridge that detaches it from the actin filaments. And lastly, ATP provides energy for the active transport of calcium at the end of muscle contraction, so that the calcium can return to the sarcoplasmic reticulum, ready for another round of muscle contraction. As an assignment, I'd like you to differentiate the slight differences of mechanisms of contraction between skeletal muscle, cardiac muscle, and smooth muscle according to the stimulus type, the second messenger involved, the source of calcium, the calcium receptor or the calcium binding protein, and the duration of contraction period. Submit this in PDF short size document in groups. Thank you for listening.